From cell culture to polishing, numerous steps are required to produce a viral vector, even before purification and product commercialization. While the manufacturing process for viral vectors follows many of the same steps as a monoclonal antibody workflow, a viral vector is much larger and has a dramatically different structure than a monoclonal antibody, given it is meant to be infectious. Complex purification steps, more difficult scale-up, contamination risk, meeting BSL-2 standards, and large-scale manufacturing capacity all have to be addressed to ensure safe and efficient production of flexible viral vectors for gene therapy. So, what can you do differently to simplify the approach and produce the best possible viral vector? For starters, it's time to best foresee manufacturing risks up front versus at the preclinical stage. Researchers often must wait 12 to 24 months to secure enough vector for their research. Availability of manufacturing facilities with a capacity to address large-scale production needs can help reduce bottlenecks that delay the start of preclinical research. An integrated end-to-end approach comprised of modular single-use technologies can also provide the flexibility that viral vector production requires. Configurable FlexFactory manufacturing process trains can help scientists better control their viral vector production and processing from multiple plasmid transfection through aseptic drug product fill, all within the same space. Using a FlexFactory train also allows more flexibility for tailored vector production and accelerating speed to market by approximately 50%. A win-win for scientists, R&D, and manufacturing stakeholders. It offers simplified logistics, on-time manufacturing capacity, performance, and operations, and low CapEx and OpEx costs. With the FlexFactory train, high-mix, efficient manufacturing is possible to advance the cell and gene therapy industry and get therapies to patients faster.